Today we're going to move on to cell membranes. This area will help us walk through questions 23, 24, and 25. From right as of now, we've just talked about how cell membranes are a part of the cell, and they define the cell, what is in the cell, what is outside of the cell, and that they are used for allowing things in and out of the cell. As we watch here in their first video, they are going to show that cell membrane is not only the phospholipids, but there's other parts that play an important role within the cell membrane.
Today I'm going to show you how to draw a phospholipid bilayer for the plasma membrane. So to start with, I'm drawing some circles across here. These are going to be the heads of some phospholipid molecules. Now I'm drawing in a protein that goes through both sides. So it's an integral protein. The second one here, it's just penetrating into one layer. It could be an integral protein, but I'll just call this one a peripheral. The next one's going to be another protein, and I'm going to add something to the top of it in a moment. Now I'm doing the heads of the other layer. Now I'm drawing in the hydrophobic tails. Try and make them about the same size and try and make them come all the way into the middle. Easier to do on paper. You could draw these as squiggly lines because they actually do have kinks in them in the molecular structure. So really it's just a representation. Spin those bits. Now I'm adding on a chain of sugars. I'm just doing them as really kind of circles hanging on here. Probably they should be hexagonal to show that they're hexose, each individual unit, but at this scale, it doesn't really matter. So labeling the integral protein, this one's actually a polytopic membrane or a trans membrane because it goes through both. This one, a peripheral membrane. Peripheral membranes can be, peripheral protein I should say, they can be just inside the membrane there or just attached to the surface. This protein's a glycoprotein. It's called a glycoprotein because it has a sugar chain on the end called an oligosaccharide. Oligo means few, saccharide means sugar. Now I'm actually going to label this as a phospholipid bilayer. And I'm going to label some of these tails as hydrophobic tails and hydrophilic head. I can also label the whole thing, the whole one of these molecules as a phospholipid. One other thing I could do is to label the cytoplasm. And then we're done.
So as he's drawn here is, you can see in the membrane that we have a couple of different proteins that are important. So we have our integral protein, we have our peripheral, and then we have our glycoprotein. We also have the phospholipids, but as he's just demonstrated that the cell membrane is not just a phospholipid bilayer. We have other very important proteins that play a role in. Now if I can move. Okay. What happens when you drop oil into water? They form these perfect circles. This is because the oil itself is hydrophobic. It doesn't want to be by the water at all. The oil droplet stays together and makes a perfect circular shape. The oil molecules are hydrophobic. Oil molecules are non-polar. Water molecules are polar. Again, they're trying to stay as far away as from the water as possible. And I'll show you how the hydrophobic ends of the phospholipid do this also. So we move on to phospholipids. They have phosphophilic head, which you can see here. This would be this part. And then we have a hydrophobic tails. The tails do not want to go by water. The hydrophobic or hydrophilic, philic meaning loving, hydro water loving, doesn't mind being by the water. So it's non-polar, polar. Water is also polar, so likes to be by. The likes like to like each other. The non-polar does not want to be by the other one. Let's clear this. Turn it off so we can move on. What happens when you add phospholipids to water is they will self-organize. They will self-orientate to keep their heads wet and their tails dry in the water. On the left, you can see that they have put all their little legs in together with the hydrophilic heads, water-loving heads on the outside. You can also see on the right, they've done the same thing, only there's so many of them that they have a little space in the middle, and it looks like a very, very crude cell. If we had some cytoplasm, DNA, and ribosomes, we would have a cell. But as you can see, we just have the membrane, and you can start to see how it forms. On the next slide here, you can see a 3D representation, again, of the two and how the phospholipids organize. Again, they're trying to keep those tails away from all the water. So as we've been talking, we talk about our cell membrane and that it's made up of phospholipids. But as the drawing in the beginning of the video showed, we do not just stay all phospholipids. We have a bunch of other proteins that are involved. We have integral proteins that are permanently embedded. They go all the way through. Some are going to be polytopic, meaning they go through both surfaces. Others are going to be monotopic, meaning they only go through either the inside or the outside membrane layer. We have peripheral. They're usually temporary. They will be there for a little bit and then leave. The importance is that integrals are permanent, peripheral are temporary. We also have glycoproteins, which he drew at the beginning, are proteins with oligosaccharides, a few saccharides. They're important for cell recognitions by immune systems and also hormone receptors. These receptors, or the recognition in the immune system, is if you have a white blood cell that is getting rid of bacteria infection, it's not really going to slow all that much down whether it's a good or a bad cell, a human cell or a bacteria cell. So it starts kind of eating everything, and what will happen is it will latch on to, let's say, a liver cell, and as it starts kind of feeling the cell, it will recognize these glycoproteins and then release the cell so that it doesn't digest it. The cell membrane also has cholesterol in it. I really love the analogy that it gives here. Think about if people were in fuzzy sweaters walking around, they'd be able to slip past each other. But if you put a couple of people with Velcro suits in, which would be the cholesterol, it really kind of makes people slow down, can't really move, and that is how cholesterol helps kind of structurize the phospholipid a little bit. There are 6 functions for these membrane proteins, and we're going to talk about them here in a little bit, and then we will be done for cell membranes. So the first one is cell adhesion. It enables cells to make tight connections to one another. The next one is channel proteins. They allow or help ions and large molecules to pass through the membrane by diffusion. Diffusion, remember, is high concentration to low concentration. So if we have a lot of potassium ions that need to get inside the cell, they may go through a channel like this. The next one is a protein pump, and it moves ions across the membrane to create and maintain concentration gradients. There is energy that is required for this. This is why it's called active transport. If you look at the pump here, you have to exert energy to get the water through, very much like the pump itself in the membrane. We have to give it energy in the form of ATP before it will let anything come across the membrane. Some other proteins are used for hormone binding sites or receptor sites, and they bind to a specific hormone that starts signaling process to change the behavior of a cell. Other proteins are used for cell-to-cell communication, neurotransmitters and synapses. So some of your nerve cells do this. They release neurotransmitters and they jump the synapse, and then they attach to the other neuron or nerve cell, and then it acts in the way it wants. The last one is immobilization proteins. These proteins inside help bind to our cytoskeleton, and that way when we need to use those proteins, they're always in the same spot. This is as far, I know it's not very in-depth about proteins, but it gives you the basics of what the cell membrane structure looks like and what its functions are, especially those proteins. Check back later. We will be doing transportation for our next video.